Hey everybody, this is Troy Hinke with Living Roots Compost Tea. I'm here at Living Web Farms to discuss assessing soil biology tonight. First off, let me explain, I've got an eye patch on tonight because I've got an eye infection and it looks pretty nasty, so I've got this on. It's also affecting my thinking a little bit because I'm dealing with being sick, so I'm going to do my best to get this information over to you, but I might just be a little discombobulated here and there. Anyway, so this class is basing information off of Meredith Lee's class a few years ago. She had done a assessing soil biology class, so I'm going to assume that most people who are watching this have a knowledge of soil food web characters, and we're going to take those characters and you're going to learn how to identify them with a microscope and use your microscope to look for them and see how your soil or compost teas are doing. So we're going to start off with soil food web organisms. I'll do a quick run through of organisms just so you know that what we're talking about, and then we'll get into how to identify soil food web organisms with the microscope, what we're going to be looking for with shapes and movement and things like that, and how to use your microscope. And then I'll be going through how to go through a slide. So when I'm starting out, what magnifications and everything I'm using to view everything in the slide and take counts and stuff. And then we're going to, after that, have some microscope time where I'll be viewing the microscope with the camera here, and we'll see what we can find, see if we find anything fun, and we'll also be taking questions and answers throughout this as people post questions to the live feed. All right. So this is the soil food web illustration. I'm sure a lot of people have seen this in books, magazines, online, or whatever. And it's basically showing on the far left-hand side of the picture how plants use the sun and photosynthesis to grow and get energy, and they are exudating chemicals into the soil, foods for microorganisms, and also providing organic matter, dead organic matter for organisms. And the first organisms that are coming along to feed on these are going to be bacteria and fungi. So we've got bacteria and fungi in the soil or compost that we're looking at, and they're going to be decomposing material. And then to cycle nutrients within the soil, we need their predators to come along. So we've got protozoa and nematodes here. So protozoa and nematodes are going to be coming along and munching on bacteria and fungi, and then larger organisms from that and other trophic levels will be coming through, munching through as well. Things like microarthropods, arthropods, worms, all cycling these nutrients in the soil. And we're going to learn how to identify all these tonight. So soil food web characters, a quick run through. We've got bacteria, we've got fungi, we've got protozoa, and protozoa are made up of amoeba, flagellates, and ciliates. And within the amoeba, we've got testate amoeba and naked amoeba. So I've got a picture coming up here of a testate amoeba. It's hard to get pictures of naked amoeba. I don't have any good photos. I can kind of explain the naked amoeba while we're looking at the picture of the testate amoeba. And then I'll be going through flagellates, ciliates, nematodes. Nematodes are going to have different food groups, so they're identified based on what they feed on. There's all kinds of different nematodes in the world. Most people know nematodes. They get a bad name because of root-feeding nematodes, which are a parasite, but there are actually more bacterial-feeding nematodes than anything. And in the whole world, the thing with the most, there's more organisms than any in the world of nematodes, is what I'm trying to get across. More than any type of bird or mammal or anything, there's more nematodes than anything. So among the nematodes, there's bacterial-feeding nematodes, fungal-feeding nematodes, predatory nematodes, and root feeders, or the parasites. And then we might also see some microarthropods, and I've got a good picture of that coming up as well. Other things that we might see, I meant to fill this list out a little bit more. We're probably going to see some humic and fulvic acids within the aggregates of organic matter. We may see some cellulose if anybody's thrown some paper into the compost pile. We might see rotifers. There's a number of things that we could see, but we'll stick to the basics here. So bacteria and fungi. when I'm going through a sample and looking for bacteria and fungi, there's going to be tons of bacteria. The shapes of the bacteria are going to be cocci or round or rod shaped, which come in different types of rods or rectangular shapes. Bacteria can also come in spiral shaped, but the spiral shaped ones that we're going to be seeing, we'll hopefully not be seeing any spiral shaped bacteria. If you do, they're generally some type of spirillum, which are indicative of anaerobic conditions. So here we've got a good fungal strand. When you're looking through a sample, you're going to see roots, plant matter, fungi, and different things. The best way to determine if you've got a fungal strand is because you're going to have uniform diameter throughout the entire strand, or at least for the majority of it. If you've got some parts that are just starting to grow, they're also going to be uniform in diameter. They may be smaller in diameter, but they're going to be uniform. They're not going to go from larger to small or anything like that. Generally, a root or root hairs or things like that are going to have a shape that's going to decrease in size. So you want to have uniform width. You want to have cell walls. So there's a cell wall here. I got out of the screen there. There's another cell wall here. Thanks. Right there's a cell wall. I did it again. There's another cell wall up there. So
give you a little more room.
Cool. There you go. All of the cross sections here is a septum, or indicating a cell wall. And you're going to have uniform cell walls as well, so they should be the same size as you go along the fungal hyphae. And then as far as bacteria, it's hard to see some bacteria in this. You've got some specks right here that are easy to see, but basically, well, also here, so this is yellow color. We've got fulvic acid inside the aggregate here, and there's bacteria that is just all surrounding there. So all those tiny little dots, there's so many that it just kind of fades into the picture, but those are bacteria. And then protozoa. This right here is a testate amoeba. So I like to say it looks like an olive shape. You've got the oval. Right down here is the mouth section. And that is a test or a shell that the amoeba goes into. Generally, when I'm looking at a sample, from shaking the sample or moving it around, they will draw themselves back into their shell, but there's a body in the shell that can sometimes come out or will come out of the shell to feed and eat on bacteria. And you can see inside there's bacteria. Some of it's behind the testate amoeba, but some of it is in the testate amoeba. That's bacteria that amoeba has eaten. Can one of you all get the, oh, it's right here. Do you have a marker for that too? Yeah. I'm going to go through the characters, and then we'll go through how to identify them, like as far as movement and stuff like that. So with nematodes, we've got bacterial feeders, fungal feeders, predatory root feeders, and then there's also omnivores. So here's a good picture of the mouth parts. When we're looking at nematodes, we want to identify them by the mouth parts, and so you have to usually get them to slow down or kill them on the slide to get it to where you can see them. Otherwise, nematodes are wriggling like a snake and just moving all kinds of crazy ways. So the first one, we've got a bacterial feeder. The mouth parts on bacterial feeder are generally going to be rather simple. Their lips, they can have some ornate lips. I'm trying to point at the one far over here. But it's just...
If you scoot more to over here, maybe you can reach further.
Cool, thanks. So we've generally got a plain mouth, and then with the fungal feeder here, I want to note the stylet here, and this is like a spear. It's called a stylet, and they'll stab that. The fungal feeders will stab that into a fungal cell and suck out the nutrients. The same thing happens with a root feeder here. So with root feeders, you've got a stylet. They stab that into a root and suck out the nutrients. With to determine the difference between a root feeder and a fungal feeder here, if you follow this stylet down to the bottom, you've got two little knobs or nodes there, and those two you're always going to have on root feeders at the end of the base of that stylet, whereas the fungal feeder does not have that. And then, so we've got bacterial fungi, root feeder, and predatory nematode. Predatory nematodes are going to eat other nematodes. Generally, they'll search out root feeders. They also are going to eat other organisms like protozoa and fungi. They'll chew through that. To determine the identification of a predatory nematode, there's a little tooth, and that can be really hard to see with the microscope. You really got to use your shadowing and things like that to see what you're looking at, but you're going to have a tooth, that black part that's sticking out there. And then with omnivores, I generally am not, honestly, have never, I think maybe I've seen one omnivore in all my experience over the past 11 years. And sorry, I'm still getting used to the green screen. They are going to be a bit larger in size, and omnivores are going to eat other organisms, other soil food web organisms as well. So here's a picture of a full nematode. They look like worms or snakes. They wriggle through the soil, wriggle through samples. They've got pointy tails. They can be long and really pointy or short and stubby. This is most likely a bacterial feeder. And then we're looking at the mouth parts here to determine what kind of feeding group this is from. And there's no tooth, there's no stylet, so this is going to be a bacterial feeder. And then I've got a picture of a microarthropod. This is a good capture. It's hard to capture microarthropods and you don't see them a lot, or you might actually see just little parts of microarthropods that have gotten ripped apart in the soil. All right, so as far as checking these things out in the soil, oops, sorry. Is it going to be best to leave it up there? And
Yeah, I was just going to turn the camera.
okay. Sorry, everybody.
All right.
Ready? So we've got protozoa. We've got the testate amoeba that we'll be searching for. Sometimes they kind of look more like a balloon. Sorry.
Got it.
Okay.
You're good?
Yeah, we're zooming.
Okay. Sometimes they look more like a balloon. Sometimes you'll have that, and then they'll have come out of their test. And then, so this is the testate amoeba. We've also got naked amoeba. Naked amoeba can be basically any shape. They look like a blob from the movie The Blob. And they use a pseudopod. So one of these parts is going to be their pseudopod or arm that they reach out and grab hold and pull themselves across the surface. So this will reach across and pull themselves that way. When you're searching through the microscope for different types of amoeba, the naked amoeba can be very hard to spot. They look like kind of a little slime spot or like snot, basically, on the slide. So it looks just like an out of focus section a lot of the times, and you might easily go glance over them and totally miss them. So, all right. Protozoa is the category we're in. These right here are amoebas. Now we've got flagellates. So with flagellates, you've got a small being with a flagellum, which is a tail that they use in a whip-like manner. Think of a sperm that's got a tail, and they use that to swim. Some of them also use that to collect food sources. So you'll see them using this tail to move around and grab hold of bacteria and pull it into their mouth. So the reason I'm using this tablet is to show you the movement of ciliates and flagellates because it's hard to capture on a camera. So with a flagellate, You may see a shape like this. They're generally going to be in the slide. They're going to be not too much larger than little spots of bacteria. They're going to be maybe 10 times larger than that to 20 times larger. But you're going to see, you're basically going to be looking for movement in the microscope. You're going to see these little beings that kind of hover around like this, and then they'll kind of bumble around like this, and then they'll bumble over here, and they'll bumble over here, and they may go out of your view, but they move just kind of bumbling is the best way to describe the way that they move around. They're getting some bacteria, looking for bacteria, they'll move over here, and they kind of do these little circle movements generally. They don't go in straight lines usually. So protozoa, flagellates, and ciliates. We're hoping to see protozoa and flagellates. Ciliates are a sign of low oxygen or anaerobic conditions, so we're not wanting to see ciliates, but that can show us that we have a sample that is having some issues. So with ciliates are generally going to be much larger than flagellates. There are some species of larger flagellates that look similar in size to the smallest ciliates, but generally ciliates are going to be rather large in size. Ciliates can kind of come in any shapes. I'm just going to draw an oval shape, and then they are surrounded by little hairs called cilia. And usually you're not going to even see these cilia because of the movement of the ciliate. But the movement of these through your sample, you'll be viewing, so this is the square, you'll be viewing that in your microscope, and all of a sudden you'll have just something zoom across, and you saw the movement, but you didn't see what it was. That's most likely 99% of the time going to be a ciliate. They zoom across in straight lines. We'll go through the magnifications in a few minutes, but we're generally looking at everything at 400 times magnification. So if you see something zoom across there, then you'd want to flip back to 100 times magnification or 40 times magnification so you can see more of your slide. And then you'll see when you zoom out to a bigger or smaller magnification, less magnification, then you'll see this thing that's zooming here, zooming there, zooming there. And you want to, just a few of those are fine. If you've got more than 2 or 3 in a sample, then you're most likely having anaerobic conditions in your compost pile or soils. So those are things that we are not wanting to see. I'm trying to think if there's anything else that I need to use this for. Yeah, so basically when I'm going through a slide, I'm looking for shapes and I'm looking for movement. I'm looking for those uniform diameter hyphae, fungal hyphae with cell walls. So I'm looking for branch shapes as I'm going through the slide. And then I'm looking for movement. So I'm looking for this zooming movement, I'm looking for bumbling movement, or I'm looking for nematodes that are going to be generally wriggling wildly, or just the shape of a nematode. So while you're looking through the sample, I'm going to look for the shape of something that looks like a worm with a pointy tail. Sometimes they're going to be just kind of still, and they may be hidden in some soil aggregate or whatever aggregates in the sample, and you really got to hunt. But generally, they're going to really be moving, and it's going to be hard to miss them. All right. We'll go back over here. So these are 15 milliliter centrifuge tubes, and I like to fill them up with water first. If you're putting your solids in there first and trying to get an even 1 gram or 1 milliliter of material, you're going to have air pockets if you put the solids in there first. So I like to fill it up. We're going to do a 1 to 10 dilution. So you want to have 9 parts water and 1 part of your solid like compost or soil. So I've got that one filled up to 9, and then I put 1 part of, this is worm compost, I believe, so it's filled up to 10 now. And then I've got other ones filled up here to 9 parts. So we're going to do what's called making dilutions. So this is a 1 to 10 dilution. I do most counts for all protozoa, fungi, and nematodes at a 1 to 10 dilution. And then I will take this and I'll take 1 milliliter of this and add it to the 9 milliliters of this. That makes a 1 to 100 dilution. And for counting bacteria, that's generally not enough. You still usually have a whole lot of bacteria. It makes it a lot easier to dilute down even more. So I will take the 1 to 100, once we made that, take that dilution and add 1 milliliter of that into this 9 milliliters, and that makes a 1 to 1,000 dilution. So I'm generally doing bacterial counts at a 1 to 1,000 dilution. And then as far as the microscope, we're using 10x eyepiece lenses, and then for objective lenses, we're generally only using the 4x or 40x. Sometimes I'll use a 10x when I'm looking for nematodes. But now we can get into how I go through a slide. It might be easy to use the paper again.
What's happening now?
I think it might be good to use that just to show quickly how I go through the slide. You good? All right, so you're going to use your 4x objective lens, and when you've got the 10x eyepieces, the 4 times 10 equals 40x. So at 40x, we're going to do a nematode scan. This is how I start off any count that I do. So I pick a corner, just usually dependent on which corner I'm closest to with the eyepiece. So if I'm already close to this corner, I'll start in this corner. So I put my point of view in this corner and start from there and do a nematode scan. So I'm looking for nematodes, and I slowly scan. So I'm moving my point of view up this way. So very slowly in the microscope, I'm just kind of moving up, and we can show you how I do this once we get onto the microscope camera. But yeah, moving up, looking for that movement, looking for that shape of a nematode, looking for movement, looking for that shape. So scanning all the way up to here, didn't see any. So then what I do is, this is going to be confusing. So this square is my cover slip on the microscope slide. The square that I'm about to draw represents, let's actually do this, what you're looking at through the microscope. So you want to pick something on your right-hand side here, like if you were to move to the right-hand side, you would want to pick a little aggregate or something like that, and then move your microscope over so that you know you're getting the full width over here of movement, so then you're not going back over anything you've already seen. So pick a little point of view there, and then move from here over to here, and then scan, 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 scan. You're going to just slowly move down to go as slow as you need to. If you need to look, stop, move, look, stop, move, look, stop, you can do that. You'll get to the point where you can really just slowly move and look for that, the shapes of nematodes and the movement. So go down like that, move over. So that's how I do a nematode scan. Let's say I did see some nematodes. I saw one right here, and I saw one right here. I want to note those in my memory because they're generally going to be moving so much that I'm not going to be able to stop and identify them. I'm going to have to get them to get very slow moving or die possibly by using a lighter, and we'll show you that in just a minute. So you've seen those 2 nematodes. You want to note that you have 2 nematodes, and then we're going to go on to doing other counts. So then from here, this is your slide, our slide cover. From here, you can pick, I generally pick 20 points. You can maybe start with 10. You can do up to 100. The more counts that you do, more fields that you do, the better your average and means are going to be. So pick a point here, and this is when we can maybe switch back to the microscope in just a second here. So we're going to look in that field of view, we're going to look for testate amoeba, naked amoeba, fungi, ciliates, flagellates. So we've stopped here. And we notice a fungal hyphae up here. So we can make a note of that. And then we're going to be looking for movement of flagellates. Generally, you're going to want to use your shadow feature to shadow this and make it a little bit darker, and then you can see more movement. You'll notice that if it's too light, you may miss a lot of these flagellates. That if you have flagellates, you may notice that you're missing them. And then as you use shadowing, you can see them move around. So you might see one over here, so you'll count that one. You might see one over here, so you'll count that one. So in this field of view, we had 2 flagellates and 1 fungal hyphae. And then I'm generally, to do my counts, you all at home are not going to probably be doing this, but I'll note the length of this, and I've got a spreadsheet that I can enter all this information into to determine the biomass per drop that I'm looking at. So I would enter, that's maybe 75% of that field of view, so I would note that the length of that is 0.75, and then I also want to note the width of this. So the width of the fungal hyphae that you're looking at, you can use a bacteria nearby to determine. So say you've got a couple cocci bacteria there, you can pick one that's in focus while the fungi is in focus, and by that dot, that's going to be your micrometers of width. So you're going to use the bacteria as a measurement of a micrometer is what I'm trying to get at. And I would say that that's going to be 6 to 7 of those little dots wide. So let's call it 6 micrometers wide. So then I would note 6 and then the abbreviation for micrometers. So I've got the length and that, and then I would also note that I had 2 flagellates. And I usually just write this out on a notebook where I've got like a grid of, flagellate, view 1, 3, view 4, none, whatever like that. All right, I think that we should be good for this. So again, we're looking for bacteria, fungi, protozoa, and nematodes. When we're going through a sample, we're looking for movement and shapes. A lot of people get kind of bogged down by like, what's this, what's this, what's this? And really, I'm just trying to focus on the organisms that I'm looking for. There are going to be other things in there that I kind of want to keep an eye on, but generally I'm just looking for shapes and movement. And then when we're starting off to do a count, we're doing the nematode scan, and then we're going to zoom in, and this is at 400x. We're going to go through and do, say, 20 fields of view to take counts on what we're looking at. And then we'll move over to the microscope. Do you have the hooked up? Yeah, if you want to switch it over to the camera. This slide may have dried. Yeah, this slide may have dried up a little bit. So I'm gonna.
Hold on, give me one.
Okay.
Now, Dan, you might have to set the computer to duplicate the screen or drag the microscope screen to the other window. Yeah.
Dan Hedinger, is it Hedinger?
Hedinger.
Is here to assist me. He didn't necessarily want it to be on camera, but he's doing a great job of helping out tonight. I wanted to make sure that y'all are aware of that. Did you get any questions coming through so far?
Okay.
I can answer them while we're getting this going.
So one question, wondering how to increase Brix levels naturally and how to encourage the most balanced microbiology to achieve it, and you may want to restate that.
The question was how to increase Brix levels, and the second part of the question, can you remind me?
And how to encourage the most balanced microbiology to achieve that.
The question was how to increase Brix levels in plants and how to get more biology in the soil. I'm kind of paraphrasing of how to get better biology in the soil to help plants. So the best way that I know to increase Brix is to get more biology in the soil if you're lacking biology. Having plenty of microorganisms to cycle nutrients in the soil is going to help to provide more nutrients and micronutrients and minerals to plants, which are going to help to up their Brix count.
There's another question.
Okay, cool. And then how to encourage the most balanced microbiology to achieve it. I'm reading a question at the same time. There's a second part of this question. It's about molecular weight of organisms. I can't answer that question. I'm just going to go ahead and say that. So to increase biology in the soil, there are a number of things that you can do. Get a good layer of quality compost onto the soil. That's going to help to inoculate the soil with good microorganisms from the compost. You can use compost tea or extract, which is a liquid form of compost, to get those microorganisms onto the soil. Both of those are going to be easier ways to quickly get biology. And then to maintain biology, it's good to use companion crops and cover crops so that you're feeding soil microbiology and continuously adding exudates and organic matter to the soil so that all of this biology is having food to survive and thrive, even when you may not have cash crops in the ground. All right, so let's go on to the sample here. I got to get my computer squared up so it's not all going at a wonky angle for y'all.
Make it full screen.
Okay. So it's harder to do with a camera because the camera wants to zoom in and it's hard to show doing a nematode scan. But basically, I'm just kind of doing this real slowly, looking for movement, looking for a shape of a nematode. And I'm not seeing any. But I did see some fungi before. So I usually also kind of make mental notes of, as I'm doing a nematode scan, because as you're looking through your fields of view, you may not come across fungi, but you can note that you did definitely see some fungi. And this right here looks like it might be a nematode, but I'm having a hard time seeing it in my screen. Here we go. It's too small. So it's just a piece of plant matter here that looked like it might be a nematode. Nematodes are going to always generally be clear. They're not going to really be colored. Whereas testate amoeba can be different colors. Usually all the testate amoeba, most of the testate amoeba that I see are clear or white colored. They can be yellow, pink, purple, brown, tan. You'll see them in a number of different colors, but generally, like I said, they're clearish or white in color. So you're going to see some stuff like...
Actually, I believe.
Yeah. Thanks. Here we go. So this right here looks like you might be like, is that a fungal hyphae? To me, just from having it this far out, it does not look like a fungal hyphae. But we can zoom in real quick and see if it is. I'm kind of interrupting my nematode scan, but I thought this is what we had in mind when doing the microscope classes, just trying to find some stuff to view. All right, we got a testate amoeba here. And I was just talking about colors. So right above my head, there's a brown testate amoeba. I'm going to use the mouse. Right here is a testate amoeba. Oop, I accidentally bumped the controls. It's really hard having one eye. So I'm going to shadow real quick and we'll see if maybe there are some ciliates. I'm going to make it a little bit darker here. Yeah, I'm not seeing any movement that showed, I'm sorry, I said ciliates, I meant to say flagellates. I don't mean to confuse you. All right, I'm going to go back out to my 4x objective lens, or 40 times magnification. I'm going to move through all this more quickly to see if we do have any nematodes, and then we'll come back through it. So I get to the top, and now I want to move over to the right here. Thanks. So let's, we'll pick this thing right here, and then we're going to move our point of view over till that gets on the left-hand side here, and then we're going to go back down. I know I'm moving rather quickly for the audience, that you all are not going to move this quickly, but I'm trying to do this for the purpose of the class. I can't forget to tell you guys how to deal with nematodes too, so I might do that real quick here. All right, we're going to move over here. Well, I should have 2 more passes up and down. I don't think that's anything. Are you finding anything fun?
I found a little arthropod on the last slide.
Oh, sweet.
If Dan says something, you might want to repeat it because then it won't be here.
Okay, I'm speaking to Dan at the same time here to see if he's got anything. He's viewing a microscope at the same time to see if he can find anything that's neat to look at, or something that we can use to identify organisms. There's a lot of organic matter in this sample, which is good to see. I'm going to go ahead and stop right here because it looks like a fungal hyphae that would be good to look at. Ooh. Looks like it may have been. Maybe not. It's also hard to get things, use a camera to get things into view to show other people. All right, so I can't positively identify this as a fungi, although it does look like it has uniform diameter and septa throughout here. But it's also very scrunched, and I can't positively identify it, so I can't count that. Oh, here's one for sure. See this long strand here in the middle? That's going to be that. I'm almost positive that's going to be a fungal hyphae. Yeah. And it's a nice long strand. So we've got uniform diameter, brown color. And one thing I failed to mention earlier, so when we're looking for fungi, we want to see, and I talked about the micrometers of width on a fungal hyphae. We were wanting to make counts or hoping to see fungi that are 2.5 micrometers or larger. Generally, it's been found that smaller, clear fungi are going to be what's called oomycetes, or pathogens that are going to be harmful to plants and soils. They're generally clear, thin, and they don't necessarily have uniform diameter, whereas beneficial fungi are going to be wider, like I said, 2.5 micrometers or wider, and darker in color, or at least not clear. So if we were to look at the width of this one, trying to get it into better focus here for you. It's hard to see a bacterium right next to it. Let's say this. They keep moving. There's a little dot right here that you may not be able to see, but I'm going to count that as maybe 1, 2, 3, 4, 5, 6 micrometers wide, again, similar to the last one. And let's shadow real quick, and we'll see if we have any flagellates in the background. Oh, it's already pretty shadowed. I'm just messing with my light real quick. It looked like there were some things. It looked like there might be a flagellate bumbling right near my shoulder here. It's not moving too much, but that right there, I was guessing. But it looks maybe more like just something that's buzzing with the sample. So I'm going back out to my 40x magnification now. I was at 400 magnification. I didn't mention that I'd gone down to 400 magnification. Ooh, what's this? Well, we've got, it looks like fungal hyphae behind me here. I was trying to get zoomed in on something else. So we've got 2 strands of fungal hyphae right here. Uniform diameter. It's hard to see the cell walls, but these are definitely fungi. And then above my head, I'll use the mouse. That's a testate amoeba right here. It looks like there might be more testate amoeba in here. I'm having a hard time with one eye. See? And I'm going to shadow real quick and see if we can see any flagellates. Like I said, usually I'm seeing bacterial feeding nematodes. In a sample, you want to see either bacterial feeding nematodes or other protozoa that are going to be eating bacteria so that they're cycling those nutrients. So generally, I either have more testate amoeba and flagellates or more nematodes. So there might be a few nematodes and not that many testate amoeba or anything. Or the opposite, there might be a good amount of testate amoeba and really not any nematodes. And it looks like that might be the case in this sample right here.
Bro, I've got something over there that looks like a little corn dog.
Okay.
I'm gonna get some of that compost on there.
I got to the end of my sample here, and then it's all full of stuff, and I might be starting to dry up as well. Do you have any more questions coming in? I can answer some while we're looking.
Are you using tubes? I don't know what TOUP is. Tube. I'm not sure what that is. People were kind of mentioning the redox cycle and talking about biochar and soil microbiology. So whatever you want to comment on any of those things, there's not a specific question. There's a lot of chat.
Okay. I was asking for questions from the audience, or if we had gotten any questions, and there were some different things that it sounds like it's not necessarily part of a talk tonight. I'm not sure what the question on
How pH affects things, and how when the microbiology is changing and the biomass is processing through, how the pH fluctuates and the cycles that go on.
Okay. Somebody asked about pH levels. Without getting into too much detail here, so when bacteria and fungi are decomposing things, they don't have mouths like mammals or sharks or anything like that. They have to release enzymes and acids, and then they absorb the nutrients. So when bacteria release enzymes, they're more alkaline, and when fungi release enzymes and acids, so they also release acids, which I just used the word acid, so therefore it's more acidic. So bacteria make more alkaline soils. If you've got bacterial dominant situation, it's gonna be a bit more alkaline, whereas high fungal content's gonna be more acidic. So in a forest system, you're gonna have more acidic soil. Like people talk about blueberries needing more acidic soil. That's because they come from a forest system or forest soil. When you're making compost, you really don't need to pay attention to pH when you're focusing on biology. It's part of more of the NPK paradigm. But if you are following the pH, like if you're making a compost tea and you're observing pH levels, I've had different people comment to me that they'll notice, a rise, that it'll become more alkaline in the beginning part of the brew, and then it'll level off more, and that's because you've got a real boom in the bacteria in a compost tea, and so those bacteria with the population increase are going to be releasing more alkaline enzymes that it's going to affect your pH in that way. So in the soil and in composts, it's the biology that's actually dictating the pH and the chemistry, and that's in control. It's always the biology.
Let's keep this next to you.
I think we've got an MIC in the top right corner of that slide.
Okay, cool.
It's a big giant one. I believe so.
You just check the phone.
I wasn't seeing a question there. I don't know which corner it was. What did you see in here?
Top right corner, it looks like one of those thin, wispy fungi strands. I'm doing like a 10-to-1. Is that what you said you do?
Dilution?
Yeah.
Yeah, yep. Dan had thought that he had seen a oomycetes in this sample, so we're going to look for that real quick. I don't know if I have the right corner, the correct corner.
Yeah, you can see it on 4. You can zoom way out and see it.
Way in the corner? Like directly in the corner?
It's huge.
I'm going to go down to this other corner because I think I may have moved this slide when you handed it to me.
You want to say what you're looking for?
Dan had thought he found some of the oomycetes that I was talking about that are pathogenic fungi. The clear, thin diameter fungi, fungal strands. Oh, I see what you're saying. I think those are from cleaning the slide. So if you look at these lines right here, I think that's maybe what he was referring to. This actually looks like maybe where somebody had wiped a slide. Okay. But that's not what Dan was talking about, but you might see some things like this if you zoom in too much. And like if you hadn't got the slide completely clean, it looks like that's what had happened here. I'm not finding it.
Yeah.
What is a sample of?
This is the other compost,
Okay.
the newer stuff, I believe.
You're looking at compost right now?
Yeah, so Dan had handed me a different sample. I'm just going through it real quick to see if there's anything interesting. It's a different compost that they have here on the farm. There's not quite as much aggregates and organic matter. So let me grab the mouse here. Pieces like this that are clear and jagged are going to be minerals in the soil. Looks like probably this one here too. That aggregate right where the mouse is moving a little bit, which tells me that there might be a flagellate underneath that. Otherwise, it's just me bumping the table. I can't tell. I think it's just me bumping the table. Is that the worm compost?
None of it's worm compost.
Okay. I'm going to stick another drop on here, a fresh drop. Thanks. So one other thing, I guess, yeah, you can see me. When you're using your pipette to grab something out of this, a lot of people ask this question. I generally, one thing I neglected to say, so first, before I mention that, you've got 9 parts water, 1 part solid. When you're mixing that up, to do it in a scientific manner, you want to do it the same way every time. So the way that people in the soil food web classes do it is to count to 30 and do kind of a quarter of a clock turn, so or a quarter of a circle, and do that 30 times, count to 30. I'm not going to do it the whole 30 times on camera here. And then I generally wait a few seconds after shaking it up just to kind of let things settle just a little bit. And then I'll stick my pipette halfway down the solution there, because the bottom's going to have a lot of junk and the top is going to have less substance. So in the middle there, it should be even. And then I'll draw some up, and then I'll let that sit for just a few seconds like that. And if you've got any big chunks in there, those will drop to the bottom. And I don't have any depth perception because I have one eye, so I'm hoping I don't miss this. But I'll do this a couple times to get those big chunks out of there. And then I'll come over to my slide and put a drop on there. If you've got big chunks in your pipette that are coming out into your drop on your microscope slide, it's going to impair your putting the slide cover on there, and you'll have to get that out of the way or start over. So that's the reason that I do that. And I'm just looking through this to make sure it's clean. The questions that I were seeing on there, it seems like a whole different class. I mean, that could be gone into detail in another class, not just identifying soil microorganisms. I'm going to pause real quick so that I can tell you about nematodes. So let's say we've seen a nematode in here, even though we're not seeing one. We saw one moving. We noted where it's at. If this were my slide, let's say that we saw it up here. Then we want to make sure in our memory to go back. We've done our all other counts and everything of protozoa and fungi and things like that. We'll come back. We're like, okay, that nematode was up here. So you want to move your point of view up to where the nematode was, and it's going to likely have moved. They maybe even from the time that you saw it on the right-hand side to where you did your counts, it may have swum all the way to the other side of the slide. So you want to just find your nematode again. And if it's moving to the point where you can't identify it, what you want to do is get your nematode in focus on your microscope so that it's in the right place, and then pull your slide out and take a lighter. And I don't think I have a lighter just for demonstrations, but let's pretend that my chapstick's a lighter. Okay. Thanks. So we want to light the lighter. I usually just try to count to 10, and I'll do it in increments. I'll try 10 and stick it back underneath there, and then if the nematode's still moving too much, then I'll do it again. So you don't want to hold the flame too close to the glass because you're going to get the soot from the burn stuck to the glass. You want to keep it down some. So just keep it moving, kind of keep your flame right up where the flame's touching the glass. Like that. That's about a 10 count. And then I'll put it underneath there. And let's say that the nematode has like gone from like moving like this to just kind of like slowly moving, but I still can't definitely see its head. I want to pop that back out, and I'll do it one more time. And I've had to do this up to 4 and 5 times because those little suckers don't want to die or don't want to stop moving. But you'll get it to the point where you're sticking underneath there. You may have to kind of refocus or readjust your frame, but you want to get the head of the nematode into your frame so that you can closely view it and identify it by the mouth part. I just want to make sure and remember to tell you all that while I'm going through this sample. But we can start hunting for more stuff here now. So I'm searching through these samples to try and find you all stuff to see because we're on video, but generally I would pick a point like this and just stop. Look for fungal hyphae all the way around here. I don't see any fungal hyphae. This thing is not a fungal hyphae. I can tell right here. Look for flagellates and amoeba. I'm not seeing any amoeba. I'm trying to shadow, and I'm not seeing any flagellates either. So we would note 0 fungi, 0 protozoa, or 0 amoeba, 0 flagellates. And then we would move on to another point, just like that. I'm sorry, I'm going to move down to 400x. I told you I'm not thinking very well tonight. I was up at 40x. So we're at 400x. I'm going to pick a spot, look for amoeba, flagellates, fungi. I'm playing with my shadow and light here a little bit so that it's not so bright and we can see better. Now it looks like this right here is an amoeba, but it's hard to tell the way it's in focus. And then we've got this little thing moving right here. Yeah, so I'm not seeing any fungi. I'm not seeing any flagellates. So I would note 0, 0. And then I'm trying to determine whether that is a testate amoeba. Because of the way that's butted up against her, I can't positively identify something. If you can't positively identify something, you can't note it as, you can't guess that it is that and note it. Or it's not a good practice to do that, I should say. So then, yeah, I'm looking at that, looking at a view. I flip to another one. I just happen to move this. So I'm sorry. It's really hard to use a camera because of the way that it, going from the eyepiece to the camera, it enlarges things.
Well, is this what it would look like if it was dried out? This your compost that you're looking at right now. This would be,
Yeah, it appears to be dried out.
I think I've got another resin seed and so the compost here too, right dead center in this one.
Dan was just questioning, he thought maybe this compost was too dry that we're looking at the sample of. So all of these microorganisms need moisture to survive. A lot of these microorganisms like nematodes and protozoa are actually more aquatic beings. But all of them need moisture to survive. So like as humans, we dehydrate foods to save them and preserve them. And the whole idea behind that is to keep bacteria and fungi from moving in and decomposing them and eating them and away, so before we get to eat them. In that same sense, when we're trying to build soil microorganisms and have good compost, good biology in our compost, we want to make sure that we have good moisture content. And that's the reason behind using mulches and cover crops. It's going to help to keep the soil more moist.
Couple of questions for Troy.
Yeah.
Someone is asking about pointing out humic and fulvic acids.
Oh, yeah. I said I was going to point that out, and then I kind of was looking for more for microorganisms. Someone in the audience is wondering about humic and fulvic acid, so I'd pointed out one of those aggregates that we could see humic or fulvic acid, and I'll make sure to keep an eye out for more. Right here, we're looking at 2 testate amoebas. There's a clear one and a brown one. So this one's real big. You can see the mouth part. This one, it's harder to see the mouth part, but it is a testate amoeba. And then right here in this picture, we've got this piece right here that has the yellow coloring, and that's most likely fulvic acid in the soil. Here's a big testate amoeba, big fat one. Right here, there's the mouth. There's the test or the shell. Looks like this one might be down below it to the right. Maybe not. So again, yeah, right next to the testate amoeba, this right here, you got a yellowish color in that substance. That's going to be fulvic acid. Fulvic acids are going to be golden color, a honey color. Humic acids are going to be brown.
What kind of microscope are you using?
My microscope that I currently have is a Omax brand. I think it's just omax.com or omaxmicroscopes.com. It is the model M8311, and it costs like $315 or $340. It's been going up in price. So it's the M8311 model. If you look it up for the model or SKU number, and then they have a bunch of different packages that come with different different cameras. You can get it with a carrying case. Yeah, they have just a bunch of different packages. It's a very inexpensive model. It's the one that Elaine has always recommended that's affordable for people to just start off with. I'm actually about to upgrade. It's been a long time coming that I've needed to upgrade a microscope, but and then the camera, well, this is Dan's camera. My camera was not working, but usually, just like with other cameras, the more pixels that you can get, the better your camera is going to be, and the more money you spend on your camera, the better it's going to be.
Here's another corn cob.
Okay. I'm not sure what that would be. Maybe it's a spore.
Yeah.
I passed by a spore a second ago with this, and I was trying to go back, and I was having a hard time finding it to show you guys. Edge of the slide. This is me using the shadow. I'm trying to darken it up a little bit to see if we see movement of flagellates. As far as protozoa, I usually want to see at least an average of one per field of view for good soils and compost. And I said an average, so say you have 3 in one field of view and 0 in another. I'm going to flip out here and see if I can see some more fungi somewhere else real quick. Zoom through this microscope. So here, I won't even zoom in on it. Right in the middle of the slide, you can see cells. That's going to be a piece of plant material. You can kind of see the veins and cells of the plant. I'll see if I can zoom in more. It might just make it way too much. Yeah, here we go. So this is at 400x. It's even closer when you see it through that camera, but that's going to be a piece of plant matter. Here, that big thing, that's going to be humic acid in that aggregate. You've got this dark coloring on the left-hand side, the entire left-hand side of the screen. One giant aggregate. It's got humic acid in it. So when I'm it's hard to see organisms when it comes to these big aggregates, but it pays to stop and check them out, because when you've got fungal hyphae going through soil or compost, they're attaching themselves to the organic matter to decompose it. So with that in mind, they're likely going to be really connected to aggregates that you have and bits that you have in your sample. So stopping and checking out these aggregates, even though you can't really see through them, you might see on the edges, which this one doesn't, you might see a fungal hyphae poking out one side or all sides of it. And you'll realize that you have more fungi than you had in the sample. How are we looking on time?
It's 8:20.
Okay.
And we have another question. How are you prepping your slides for this work? And please repeat the question.
How am I prepping my slides for this work? Let's see. So someone asked about slide prep. Usually, I mean, it's just a matter of taking a clean slide. So I like to, even if I start off with what looks like a clean slide, I might put a little water or rubbing alcohol on it and wipe it down with there's things called Kim wipes that are lint-free cloths that you can get from like a laboratory supply, or you can just use newspaper. That's what I like to use. Newspaper are lint-free. You can just rip them up into little pieces and use those to clean your slide covers and slides off. And then as far as prepping a slide, I didn't exactly show that anyway. This is a slide that already has slide covers on. I'm just going to pop them off and use this for an example. Clean slide. Just to reiterate for this question, I filled this up to 9 milliliters of water. I've already taken some out of here now, that's why it's down. But it was filled up to 9 milliliters of water and then 1 milliliter of the solid, which was a worm or compost sample, added to the top of that. So we had a equal full 10 parts, but we've taken some drops out. And then we shake that up 30 times like this. And usually when I'm putting my compost in there, like if you've got chunks, you don't want to just be tossing chunks in there. Like if I've got, I'll grab little bits and I'll kind of crumble them up as I'm dumping it into the water. If you pretend that the cap was off, I'll crumble those into there like that. I'm trying to break up any aggregates, any clumps, so that we're getting as much surface area as exposed as possible in the water to pull off. So basically what you're doing when I talk about compost extracts and teas and in other forms, a compost extract is when you're putting compost into water and extracting the microorganisms off of that. This is a really tiny extraction. So you're taking water and you're taking compost and you're shaking it up and you're trying to extract those microorganisms from the organic matter in the water into the water, so then we can view those microorganisms. So slide, pipette, 1 to 10 dilution. And I have one eye, so I have no depth perception. It's really hard for me to do this. I had mentioned I go down halfway, pull a little bit out. I sit here for a couple seconds and let any big chunks come to the bottom of that. Pop them out. And then put a drop, like I said, I don't have any depth perception. Put it on your slide. Squirt the rest of that back in there. Generally, I'm leaving this on my stage for my microscope instead of holding it in the air like this, because I'm just doing this for the purpose of having it on camera. Then you want to make sure that your slide cover is clean as well. Even if you're just pulling them out fresh out of the case, they'll still be dirty. Like this one could be wiped off, but I'm not going to wipe it off because I'm just putting it directly on here. So it's good to try and put them on there squarely. They're not always going to land exactly square with the slide cover. So yeah, you just let it drop down on there, and the drop of water will spread out underneath the slide cover, and then you put it underneath your microscope and check it out. And then recap for that, because that question just came through. We're going to be using the 4x objective lens and 10x ocular lens to make 40x magnification. We're going to be using that 40x to scan for nematodes at first, and then we'll go down to the 40x magnification and 40x objective lens, which equals 400 times magnification, and then we're going to be doing most of our counts and viewing at the 400 times magnification. So when we've been going through most of these slides here, we're looking at stuff at 400 times magnification. Right now, I pulled it out to 40 times magnification so I can look and see if we've got any fungal hyphae or anything to zoom in on that we can check out and identify. So far, I don't think this sample has any nematodes. We should have seen something wriggling or squirming by now. So I'm going to pull up real quick. This is the edge of my slide. And you can see this starting to happen where we've got this dark line with all this crap that's starting to get pushed together. And if we were continue to let this go, what's happening is the microscope slide is drying out. You just saw it, you can see it moving this way. The light's on, it's heating that stuff up, and it's making the microscopes, the drop in there dry out. And so that's why you have to refresh your drops, because if you leave it for so long, it's just going to dry up like that. Check out this right here. So we're going down to 400 times magnification. I think this is a big long hyphae we've got. I gotta find it. Yeah. I can't get it to zoom. Well, before I do that, it looks like there's a testate amoeba right in the center there. So in this drop of water, we also have to think where I'm using my fine focus to go up and down through the drop. Even though this is tiny and microscopic, we've got, if you could make this big, we've got a drop of water that's sitting under, my thumbs are the slide and my fingers are the slide cover. So you've got a drop in between there. So you've got this much area to, like, you know, there might be a fungal hyphae up here, but if you come down to this level, you've got it out of focus. So we're having to go focus through this entire drop to see different things in here. Here's that fungal hyphae. So like, let's do this real quick. It's not responding. Here you can't necessarily see the fungi, but if we zoom down, then it comes in focus. If you keep going, then it comes out of focus. So we're trying to get that level where that fungal hyphae is. Now this is a weird example because I said to look for a uniform diameter, and it's got some pieces that are kind of chewed up here, beaten up, but this is, I would say this is a fungal hyphae for sure. You've got cell walls in there. And so fulvic acid here, people were asking about fulvic acid. This golden color in here, that golden color that you see is going to be fulvic acid. I felt like at first we were seeing a whole lot, and then lately there's just not much coming up. Here we go. Here's a testate amoeba. Right here. So this one, I said there's some that look more like a balloon shape, more than an olive shape, and this has got that round with round shape and then a little tiny neck at the bottom, kind of like a balloon. And then I'm going to shadow a little bit and see if we can see any flagellates. I don't think there's any flagellates in there. Usually across, you'll have good averages across the slide. Like, you might stop at one and you'll have so many flagellates, and you'll go to another field of view and you'll have so many flagellates. But sometimes you might not see any, and then like you'll get to one corner of the sample, and then for some reason there's some type of food source there that there's just a bunch buzzing around. Any more questions?
Can you repeat the name of, and just repeat the question, the name of the microscope, the model, and the one that you're upgrading to?
Yeah, the microscope that I'm currently using is a Omax, and I think it's omaxmicroscope.com is the website, or Omax. Okay. And then the model number, it's model M8311, and they also kind of use that as the SKU, like if you were to search on their website. And it's a little over $300. The model I'm upgrading to, I don't remember the exact name. It's an Infinity. It's from LW Scientific. Elaine has a new soil food web school, and Keisha Wheeler had gotten a deal with, Keisha helps Elaine with this new school that she has, and she'd worked out a deal with LW Scientific microscopes for soil food web class attendees.
I4 Infinity?
Yeah, it's an I4 Infinity, yep. And then the camera that I have is like 11 years old. I don't even know if the company is still around. The camera that I currently have is a Dynolite, Dynolite. And the camera that I'm upgrading to is a really fancy, expensive camera. But what I was mentioning before was you want to, like with any other camera, the more pixels that you can get, generally you're going to have better definition and resolution. And then, what was I going to say with that too? Oh yeah, you get what you pay for. So it's usually good to spend at least $100 or $150 on a camera. I would nowadays even $200 as a minimum to get something that's decent and good. And you can also get carrying cases for your microscopes. So because I come around and do things like this, I make sure to get a carrying case for my microscope to keep it protected, since it's an investment. Did you see anything there?
Well, two things I wanted to say. I've got video of nematodes swirling around on this. If you
Oh,
want me to
cool.
show that.
Yeah.
From prior samples.
So Dan had just mentioned that he's got some videos of nematodes that are swimming on his computer that we can bring that up and show you all the movement of the nematodes and see what's going on with that. Just to have some view of nematodes.
Yeah, can you switch me off while I go to look for it? It might take me a second. Another thing here too, this is completely off topic from soil, but I'm back here looking at my hydro system, and you can see a lot of salt crystals, which might be kind of interesting.
Okay.
If you wanted to do that.
Dan's looking at a hydroponic sample that he's using. Can I say what you're using?
Yeah.
He's using human urine as a form of nitrogen in there. So he said that he's got some salts that are visible. Usually salts look like the minerals that I was pointing out. They'll just be like fragmented pieces in your sample that you can see that are clear or generally light colored. Do you have any other questions that are coming up?
Not right now. Just say farmers are rock stars.
Farmers are rock stars.
What magnification would you recommend to see bacteria for people?
The question is, what magnification would you use to see bacteria? And again, that's 400 times magnification. What I was mentioning earlier is about doing dilutions. So you could see when we're going through the sample here that there's stuff everywhere. And when it comes to bacteria, there's just like tons of bacteria to the point where it's impossible to count. So we start off with a 1 to 10 dilution where you've got 9 parts water, 1 part solid, compost or soil. And then we have other, just wanted to make sure I was still up on the camera. We fill other centrifuge tubes up to 9 parts water, and then you take this 1 to 10 dilution, take 1 milliliter out of that, add that 1 milliliter to the 9 parts, so that makes a 1 to 100 dilution, and then you'd shake that up, take 1 milliliter out of the 1 to 100 dilution and add it to this. And then once you get to that 1 to 1,000 dilution, you'll start to be able to count the bacteria a lot better. Sometimes you have so much bacteria that you even have to take it up to 1 to 10,000 dilution. But generally going to the 1 to 1,000 dilution is going to be enough. And that'll space all this bacteria out to where you can take like 1/4 of your field of view and say like, okay, I've got 5, 10, 15, 20, 25, and you'll probably count anywhere from 20 to 100 and some. Well, say you count 30 here, then you've counted 1/4 of the field. You can multiply that times 4. Instead of counting all the bacteria in your field of view, just count, a space of that and then do the multiplication. That's what I was saying. But yeah, to count bacteria, it's much easier to go up in dilutions with your centrifuge tubes so that you're counting less bacteria and you don't have all this other stuff that's blocking your view. As far as centrifuge tubes go, it's really hard to purchase these in small quantities. You generally have to purchase like 30 packs, 50 packs, 100 packs, but these are 15 mil centrifuge tubes. We're only using 10 of that, but I haven't ever found the 10, just 10. But any laboratory or scientific supply is going to have those. And then I've got plastic pipettes. You can get glass pipettes. These plastic ones, same thing. It's hard to find those in small quantities. You generally have to buy like 50 pack, 100 packs. So I've pretty much got lifetime supplies of centrifuges and centrifuge tubes and pipettes and slides. When it comes to purchasing microscope slides, these ones are from Azer Scientific that is in Pennsylvania, I believe. They're made in Germany, but it was sold in Pennsylvania. They're high quality. Again, you get what you pay for. So if you're seeing extremely cheap microscope slides, I probably wouldn't buy those. I'd probably spend a little bit more to make sure that you're getting something of quality. And those are 1 millimeter by 75 by 25. And then you got cover glasses that are what goes on that. And the size that I have here are 18, so that's the size of the square, 18 by 18 millimeter. Generally for doing these counts, either the 18, 19, or 20 millimeter size of the cover glass. And these are from Azer Scientific as well. I honestly don't know a lot of names of different types of makers of slides and slide covers and glasses and things like that. But same thing, look for scientific supply or laboratory supply. LW Scientific, which I mentioned for microscopes, they're going to have things like those on their website. AZER, Azer Scientific.
And how are you labeling and organizing your slides and samples? And
Is that a question from the? I don't really know how to answer that.
do you have any suggestions for that? That's okay. Someone said high bacteria, someone was told is good for vegetative and flowering and dominant, and to have a dominant fungal during flowering, and for vegetative growth have bacteria. What do you say about that?
So the person was mentioning bacterial dominant soils or soilless mediums, for plants in their in their vegetative stages, and then more fungi in the soil and soilless medium for flowering stages. It's actually dependent on the plant more so. So this is kind of a whole nother class as well, but it depends on succession. So early succession plants are like pioneer weeds and short grasses and things like those. Those are going to be bacterial dominant, found in bacterial dominant soils. Row crops, tomatoes, corn, peppers, and things like that. Tall grasses are going to be equal ratio of in the soil. The microbiology is going to be equal fungi to bacterial ratio. And then as you get into more shrubs, trees, forest systems, you're going to move into more fungal dominant soils. So with the plant that I believe that the question was asked for, you would actually want close to equal bacterial to fungi ratio, or even more fungi. So there had been a question asked earlier, and I didn't. I could have added a few more answers to that. There was a question earlier about how can you get more biology into soils or whatever growing medium that you're using, and I'd mentioned using compost or compost teas, as well as using cover crops to keep those things alive. You can also purchase mycorrhizal spores. So when you're sowing seeds or doing starts, you can add either a slurry or powder form of mycorrhizal spores, where you're soaking your seeds or dipping transplant into mycorrhizal spores. The mycorrhizal spores will make a connection with the root, and then you've got, hopefully they will grow with the plant, and you have the advantage of having that mycorrhizal fungi aid in bringing nutrients, minerals, and water to your plants. One other thing that you can do is to, if you're using cover crops or planting leguminous plants, you can use nitrogen fixing bacteria that can be purchased in a powder form that you can soak seeds in, either the powder or a liquid form. And then these nitrogen fixing bacteria are going to work with the leguminous plants, and they form nodules on the root system where the bacteria infect the root and are grabbing atmospheric nitrogen and converting it to ammonia, so that the plant can then make use of that nitrogen, and they're pulling nitrogen from the air to make use for the plant. So yeah, compost tea, cover crops, mycorrhizal spores, nitrogen fixing bacteria. I can't think of anything right off the top of my head. This thing's starting to bother me. I either need to take a break or cut it. Okay.
Maybe 2 small questions and then we're almost done.
That sounds good, yeah.
So, what kind of water do you use for your solution? Is there any particular thing you recommend?
For taking samples, the question was what kind of water that I use. When it comes to, this isn't part of the question, but like when it comes to making teas or extracts, I usually talk about taking up chloramine or chlorine using humic acid, or trying to not have chlorine or chloramine by using rainwater. When we're just doing small things like this, it doesn't necessarily matter. Usually, if you've got chlorine in your water, it's not like you're going to put a sample into the water and all that biology is going to be like, oh my God, I'm dying. You're still going to be able to use that. So I usually use filtered water or distilled water. I drink filtered water at home, so I just use that filtered water. But yeah, tap water is fine too, for the water for your solution for using a microscope.
Okay, and one final question. Can you have too much biology, and what would you recommend that?
The question was, can you have too much biology? I cannot think of a scenario where you would have too much biology, just possibly the wrong biology. So I was just talking about plants and succession before, and bacterial dominant soils, fungal dominant soils are equal ratios. So you could have the wrong biology, and that's what we're trying to remediate through compost, compost teas, and other things is to get this biology, get your soils to the correct biology. Yeah. So most soils, because of our current management practices and management practices since plowing began, plowing, chemical usage, heavy machinery and compaction, all of these are going to, I shouldn't say are going to, all these have the possibility of having negative effects on soil microorganisms. When it comes to plowing and chemicals, I would say definitely they're going to have impacts on soil microorganisms. So if this is your soil profile and you've got these strands of fungi, if you can picture small threads like sewing threads just running back and forth through this soil profile, if you come in there with a broad fork or a plow or something like that and just completely rip that up, you're tearing all of those threads and all those microorganisms. It would be like if a large city were to have a hurricane come through, everything's just getting mixed up and killed and jumbled. So, with fungi through the soil, especially mycorrhizal fungi, you've got all these connections and interconnections, basically like basically like humans having highways and power lines and this grid that we're on. These soil microorganisms are forming their own cities and metropolises of connections, and when you're using a plow or something like that, you're coming in and basically ripping apart the city and making them all start over to the point where you're kind of killing a lot of the fungi and getting mostly just bacteria that are coming back, which I just mentioned succession just a few minutes ago. If you have bacterial dominant soils, that's where weeds are going to grow. So through chemical usage and tillage, you're actually making your soils more likely to grow more weeds than have less weeds. Other than that, if you want, can you transfer over to the nematode picture on his camera? Dan had brought up the picture of a nematode so that you guys, is that a video?
Video.
Okay.
I checked the date though, and I wanted to make it clear that this was about a year ago. This is the same sample.
Oh, okay. That's funny. So this is from the same sample that we were just looking at, but he was mentioning that he thought the compost had dried out. So a year ago, he had viewed this same sample or same material and found this giant nematode, which I don't know how to make the screen move on his camera, or on his computer, but it looks like it's really big. It's hard to tell. Can you zoom in at all? On the camera, it's like not on its head part. Can you get to the head part? There you go.
Let's see.
We're going to check this out real quick, and then we're going to wrap up for the night because it's getting close to 9 o'clock. So yeah, this is a picture that was taken, so we can't change the focus of this at all. But I'm not seeing a stylet. I'm not seeing a tooth, and so I'm left to figure that this is a bacterial feeding nematode. But yeah, so I was just going to mention too with sizes, it's hard for me to mention sizes with this because everything's microscopic and you may not be used to this, but once you get used to looking at things, you're going to see some smaller nematodes and you're going to see various sizes of the nematodes. So usually bacterial feeding nematodes, they're just eating bacteria, they're going to be smaller in size, where if you've got predatory nematodes, which are eating other nematodes and larger things, they're going to be larger in size. So predatory nematodes are usually 2 to 3 times a lot of the bacterial feeding nematodes that I see. But again, bacterial feeding nematodes can come in different sizes too. But yeah, if you've got a sample where you're like seeing one nematode over here that's real tiny, and then on the other side you've got this one that looks really big, they're likely different types of nematodes from different feeding groups. Other than that, hopefully we've covered everything. Hopefully I went through the details. I'm a bit out of it today just because I'm dealing with this eye infection. I hope I gave you as much information as I could. We may be coming back at a future time for a similar class like this we talked about. I don't know that for sure. Maybe I shouldn't have even mentioned it, but I just wanted to say that it's a possibility. Once I get this new microscope, we can come back with an even better microscope and camera so we can get better videos and show you all better stuff. But other than that, And I hope you enjoyed tonight. Thank you for tuning in. Thank you to Living Web Farms for inviting me, and have a good evening. Happy St. Patrick's Day.
We'll see you at Organic Grower School.
Yeah, we'll see you at Organic Grower School this weekend. I hope you signed up for that. See you later. Have a good night.